Hello everybody, I'm Samin Abolhassanzadeh. I'm as a scientist in Marriott Nutrition Science in Ireland. I'm very pleased to give me this opportunity to share my experience with you. The topic of my presentation is production of recombinant protein that inhibits cancer stem cells. This presentation is divided 3 stages: introduction, methods, main result, and conclusion. Step 1. Introduction. The present invention is in the field of developing a new therapeutic approach in the field of medicine and molecular biology, focusing on the production of soluble FZD7 receptor FZD7 CRD as an inhibitory tool in the β-catenin signaling pathway. The signaling pathway is known as one of the key pathways in regulating cellular processes such as proliferation, differentiation, and self-renewal of cancer cells, especially cancer stem cells (CSC), cancer stem cells (CSCS), pose serious challenges in cancer treatment due to their unique abilities, including metastasis, resurgence, heterogeneity, and resistance to common treatments such as chemotherapy and radiotherapy. Therefore, inhibition of these cells through distribution of vital signaling pathways such as β-catenin can be proposed as an effective strategy in fighting cancer. Step 2. Method. In this invention, the soluble FZD7 CRD receptor, which specifically targets the extracellular domain of the FZD7 receptor, has been produced by recombinant method. This soluble receptor is able to bind competitively with WNT ligands, which play an essential role in the activation of the WNT beta-catenin pathway and prevent the binding of these ligands to FZD7 cell surface receptors. This mechanism can lead to the effective inhibition of WNT signaling and, as a result, prevent the formation and maintenance of cancer stem cells. The application of this invention, including the treatment of various types of cancer such as breast cancer, digestive system cancer, lung cancer, and skin cancer. Steps taken in the synthesis and implementation of recombinant CRD FZD7. Number 1. Amplification of FZD7 extracellular domain gene sequence by polymerase chain reaction and gel purification. Number 2. Insertion of FZD7 fragment in T vector plasmid PTG19 and production of PTG19 TFZD7 construct. Number 3. Transformation of PTG19 TFZD7 construct in E. coli strain top 10. Conformation of FZD7 fragment in plasmid vector by PCR colony. Sequencing, restriction, endonuclease digestion methods. Number 5. Subcloning of FZD7 receptor extracellular domain nucleotide sequence in PET28A plus expression plasmid. Number 6. Transform the construct FZD7 PET28 28A plus in E. coli strain shuffle. Number 7. Induction of recombinant protein expression. Number 8. Confirmation of protein expression by SDS, PAGE, and Western blot methods. In the results section, we have several figures and charts. We discuss them. Figure 1 and 2, the effect of FZD7 CRD on the proliferation rate of gastric cancer cell line AGS and colon cancer SW480 at concentration of 50 microgram per milliliter, 25 microgram per milliliter, 12.5 microgram per milliliter, 6.25 microgram per milliliter, 3.12 microgram per milliliter, and microgram milliliter 1.56 was measured by MTT method after 72 hours of treatment. Figure 1. Inhibitory effect of FZD7 CRD including apoptosis of AGS, gastric cancer cell lines. Figure 2. Inhibitory effect of FZD7 CRD in including apoptosis of SW480 colon cancer. Figure 3. The effect of FZD7 CRD in inducing apoptosis of AGS gastric cancer cell line at a concentration of 25 microgram per milliliter after 72 hours of treatment was measured by annexing per pi method. Figure 4. The effect of FZD7 CRD in inducing apoptosis of gastric cancer cell line SW480 at a concentration of 20 microgram per milliliter after 72 hours of treatment was measured by annexing per pi method. Eventually, we have a conclusion step. Due to the important role of the FZD7 receptor in the progression and metastasis of tumors, targeting this receptor can help improve the effectiveness of anti-cancer treatments and reduce the likelihood of relapse and drug resistance. This innovation can contribute to the development of targeted therapies and reduce the side effects associated with cancer treatments and can be considered as a promising candidate in the field of cancer treatment. Advantages of this invention compared to previous inventions. Number 1. Specific inhibition of binding of WNT ligands to the FZD7 receptor. FZD7 CRD directly interacts with the FZD7 receptor at the cellular level and prevents the binding of WNT ligands to this receptor. This specific inhibition deactivates the WNT signaling and prevents the activation of processes that lead to the growth and survival of cancer stem cells. Number 2. Intrinsic protein by simulation minimal immune reactions. Number 3. Reducing the number of the cancer stem cells, migration and metastasis. Number 4. Reduction of side effects caused by chemical drugs due to specific function. Number 5. Increasing the survival of proteins. Number 6. Reducing treatment costs due to the use of chemical drugs. Number 7. Lower production costs than monoclonal antibodies. These benefits indicate the high potential of FZD7 CRD as a novel and effective cancer treatment method that can help improve treatment outcomes and reduce treatment-related costs.